talked about DNA, how it was discovered, how it replicates. Now we're going to talk about RNA and how we make that through transcription and translation. All right, so remember, first off, RNA is ribose. Okay, it has that sugar ribose. That's why it's RNA. So ribose nucleic acid. It contains uracil instead of thymine, and it's single-stranded. DNA is double-stranded. So there's your three main differences between RNA and DNA. All right, so RNA, there's three different types. We'll see that. There's messenger RNA, which is the long strand that we will see, and we make it from our DNA. Our ribosomal RNA, which is basically your ribosome. And then your transfer RNA, which are small 3 segments of nucleotides. All right, and they help make our enzymes, because that's what we've been talking about. We've been talking about enzymes, right? Okay, so we have those 3. Messenger RNA is the one that we want to focus on most of the time. This one, it'll help us to make our enzymes.
I don't see it on the photo, by the way.
I'll help you find it here in a second. Messenger RNA. All right, like I said, it's a long strand. Okay, it's going to slide into this little gap on our rRNA, and in these little segments right here, our transfer RNA brings in our peptides, our amino acids. That way we can make our proteins. All right, so when we start talking about our enzymes at the beginning of this chapter for A&P, that's how we're making them right there. That's why we're discussing DNA right now. See, when we talk about it in the A&P notes, it doesn't go into as much detail. That's why we're doing it now. Skip that. So that first process is transcription. All right, transcription, we're taking transcription, we're taking DNA, we're separating it, and instead of using DNA polymerase to make a new strand of DNA, we're gonna use RNA polymerase to make an RNA strand. All right? But we're just making one. It's just picking one side. Now it knows where it needs to make that segment from. Okay, so it's just going to go down this line until it gets all that it needs. Then it'll break off, and this DNA strand will come back together. All right, so that's what our RNA polymerase is doing. Okay, it's going to use that DNA code to make that RNA. It's going to transcribe it. You guys know what the word transcribe means?
Right.
What?
Right.
Right. Transcribe from one language to another. Okay, so we're using one code to make another. So remember, I told you at the very beginning, our DNA in every single cell is the same. Only certain genes are turned on. So our DNA, every single cell has the same DNA. It has the same code to make all of our enzymes, to make our messenger RNA. All right, so we're gonna have what's called introns and exons. I know it's kind of weird because you would think introns would be the ones that would stay. They're actually the ones that get the boot. Lane? Okay. So introns are actually the ones that get the boot. To help you remember, introns are no longer in. Exons continue to exist. All right. So there are segments of that messenger RNA that we don't want. So we're going to take like scissors and cut it out. And the segments that we do want, they're going to fuse together. And that's what's going to be left. That's the exons. That's what we want to leave behind. All right? So that's taken away from our messenger RNA. So essentially what happens is we have our strand of messenger RNA. If we don't want the segment between my fingers, the segments that are still there will come together. It'll cut that segment out and fuse those two together. That's essentially what's happening. All right, so to try to understand this code even more, scientists, they did experiments and decided that DNA was a 3-base code. A 3-base code, meaning 3 nucleotides stood for something specific. All right, you guys remember from biology what the start codon was? So you 3, you've had biology 3 years ago, so that might not be easier for you. AUG. It's on your paper too. Yeah. Sean had it last year. Blaine had it 2 years ago, so they were more likely to remember. AUG is the start codon. All right, meaning that's what's going to start making a protein. All right, so it's that 3 base code, a codon. Okay, so if you look at 12.3 study guide, it has them all on there. It's kind of hard to see. Look them up on the internet. Okay, just type in RNA codon sheet. It will show you what you need. All right. Also in the PowerPoint. So right here, AUG, methionine. So every single enzyme that we have, every single protein that we have starts with methionine. We have our stop codons up here. Okay, so we have 1 start and we have 3 stops. Okay, so translation, so first we had transcription, now we have translation. All right, we need to translate it. So we're gonna take these codons to read our messenger RNA. We have what's called anticodons. All right, anticodons. They are complementary to the codons. Your transfer RNA, so we talked about your messenger RNA is that big strand. The ribosomal RNA is the ribosome that it fits in, and their transfer RNA brings stuff in. Our transfer RNA has our anticodon. So, you guys remember what pairs with A in RNA?
T.
U, because there's no T in DNA, remember? You have yourself. So, U, what pairs with U?
G. No.
A.
A.
What pairs with G? C.
C.
So UAC would be the anticodon. That's what's on the transfer RNA. All right? If you're confused by that, you'll get plenty of practice, especially with the assignment. You guys are confused by something on that, I will help you with it. All right? But here we have our messenger RNA, this green ribbon. It's going through our ribosomal RNA. Our transfer RNA is coming in, sets in right here, moves to this middle one, and it adds its peptide to it, its amino acid, its protein, to make that protein. All right, if you look, AAA on that transfer RNA is the anticodon because it matches with the codon, which is UUU. When it moves to the 3rd section, it's being kicked out because it's already left behind its peptide. And that's how we make our proteins. That's how we make our enzymes that we need. So remember, they're gonna fold, all right, to make that shape that they wanna make, all right, to conform. Okay, so remember, DNA is vast, it's long. Certain genes are turned on while others are turned off. One gene, one enzyme. So one specific gene on our DNA codes for one specific enzyme. So that's why some cells are specific. All right, that's why our liver cells, some of our kidney cells have peroxisomes. Peroxisomes help detoxify alcohol and get rid of toxins, drugs. Okay, they have specific genes turned on to make those. All right, so they're gonna make specific proteins. Are there any questions over that? Okay, so you got the 12.3 study guide. Told you that A&P DNA test right there, it's not really a test, it's just gonna be an assignment for you. Work on that. You can find the answers in the biology textbook or the A&P textbook.
Using this on the test.